Okay, are there any questions?
No questions, but we can hear you.
Okay, great. All right, you should be seeing my screen. Let me see if I can close that. So today we're going to be discussing the Lab 7 throat and urine cultures. You're not really going to be working on your unknown works today. You won't get anything to do until next Tuesday when I send you the first results. Let me show you that we're actually way behind in our lecture. We still have about 20 slides of chapter 5 to finish, and we're supposed to be starting chapter 6 today. So we're way behind schedule. I don't think I can finish chapter 5 and chapter 6 on our next lesson. Anyways, let's get on to lab 7. Any questions about anything before we begin? Lab 7 is throat and urine cultures. There is some reading that you should do before you begin the lab. You should know about the oxidase test and the catalase test. And you should know about the coagulase test, but it tells you to see the text at the end of this lab module for learning about that. And you should know about the microbiota in the nose, the throat, and the mouth. And actually, it's only on page 392. And then there's some information about beta hemolytic streptococci and Haemophilus, which is at the text at the end of this lab module. In today's lab, there are some supplemental material, just video clips. There's no virtual lab. We had one, but it no longer runs because you need Flash to run it. Upon successful completion of this lab, you should be able to explain how to properly collect throat and urine specimens for routine culture. You should be able to list common bacterial pathogens and how they're isolated from throat and urine cultures. Be able to know the culture media used to cultivate these pathogens. Be able to recognize which tests are used to identify throat and urine pathogens, and understand the principles and how to interpret the following tests. You should know the catalase test, the coagulase test, and the oxidase test. We will talk about the, I think it's XV test, but maybe it's 15, I don't know, factor testing, but you're not going to be quizzed on it. So you don't really need to understand it or know it, okay? Any questions about that? All right, once a specimen is received by a lab, they are logged and processed in a timely fashion. Specific culture media are used and inoculated and then incubated depending on what the specimen is. For throat and urine specimens, we have specific culture media to inoculate throat and urine specimens. Below is an overview of the culture media used and the common pathogens that are frequently isolated from throat and urine cultures. For throat specimens, we mainly use blood agar. The main culprit is Streptococcus pyogenes, which causes strep throat. I think it causes scarlet fever. That's such an old disease, I don't remember, but I think it's strep throat or Streptococcus pyogenes. And it used to cause a disease of people's hearts. I forgot what that disease was called. What happened was people would get a Streptococcus pyogenes infection, and then they get strep throat, and they wouldn't be able to throw it off. That's a problem with strep throat. You can't really throw it off. And then it just gets worse and worse, and then it invades your system and causes all sorts of other diseases. Those diseases used to be common before the advent of antibiotics. But now, because of antibiotics, those diseases are not very common. Okay. And one of them was scarlet fever. I don't remember what they were. And even my generation didn't get those diseases. But there was an instructor who was older than I, and he would ask the students in the class, how many of you gotten these diseases? And he's the only one who rose his hand for all of those diseases. They were very common before the antibiotics were developed, but they're not common today. The other common throat culture media is chocolate agar, and it's specifically used to grow Haemophilus influenzae, which is a disease seen in young children and in infants. Okay, Haemophilus influenzae is not usually seen in adults, but it is common in young children and infants. And it doesn't grow on blood agar, you have to grow it on chocolate agar. Both of these organisms, Streptococcus pyogenes and Haemophilus influenzae, are fastidious. They will not grow on a general purpose media. They have to grow on an enriched media. For growing urine specimens, we use blood agar and either McConkey's agar or EMB agar. It can be one or the other, McConkey's or EMB. You grow blood agar to grow essentially everything, both gram positive and gram negatives, but oftentimes gram negatives cause the urine pathogens. And that's why we grow urine pathogens both on blood agar and either McConkey's or EMB. Both McConkey's and EMB select for gram negatives. Some common bacterial pathogens isolated in urine cultures include Streptococcus and Enterococcus and Staphylococcus. All of these are gram positive, and they will not grow on McConkey's or EMB. And then members of the Enterobacteriaceae family, especially E. coli, that's the #1 urinary tract infection, and then Pseudomonas species. Both Pseudomonas and Enterobacteriaceae are gram negative. Any question about any of that? All right, for throat cultures, if you see white patches on inflamed tonsils and then the back of the throat, you can, that is indicative of a bacterial infection. If there are white patches are missing and it's just a red inflamed throat, it's usually a viral infection. But if you see white patches, it's usually a bacterial infection. The most common isolated bacterial pathogen in the throat is Streptococcus pyogenes, and that is also called group A strep or gas. And I don't use these two terminologies. I'll use Streptococcus pyogenes where you're naming the species. But in this lab module, they use both of these. And so you have to learn them for the lab module. Strep throat may be diagnosed in one of two ways. And we're going to only talk about swabbing the back of the throat or the tonsils where there's redness and or white patches. So you touch with your swab the back of the throat or the tonsils. You do not touch the sides of the mouth, sides of the cheek. You do not touch the tongue. You don't touch the top of the mouth either. And usually the tongue depressor and the swab are used. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy. because as I mentioned, Streptococcus pyogenes is not a bacteria that we can easily throw off. In fact, generally, most people cannot throw off Streptococcus pyogenes. Since Streptococcus pyogenes is fastidious, it is usually grown on blood agar. That would be a tryptic soy agar supplemented with sheep's blood. You then examine the plate for small, clear colonies surrounded by a complete zone of hemolysis. That's called beta hemolysis. And that will likely be Streptococcus pyogenes. However, if you have a blood agar plate showing small colonies with complete hemolysis, sorry, my mouse wasn't working, shown here, there are two tests you need to run to prove that the infection is Streptococcus pyogenes. The two tests are using cadlase and bacitracin susceptibility. So cadlase is where the bacteria produces the enzyme cadlase, and it breaks down hydrogen peroxide, H2O2, into water and oxygen. And the oxygen produces bubbling. So you look at the bubbles, and then you say cadlase is present. Most cells that grow in the air have cadlase, but not all of them do. And Streptococcus pyogenes is one that is missing cadlase. Only Streptococcus and Enterococcus species test negative for cadlase production. So this would be a negative result, and this would be a positive result. Something like Staphylococcus aureus, which can cause a throat infection, although it's more likely to cause a skin infection, but can cause a throat infection, would be positive. Streptococcus pyogenes would be negative for the cadlase test. The other test to prove that it's Streptococcus pyogenes is to plate the bacteria in a swab, in a large swab, a large area of the plate, and then put on a disc containing the antibiotic bacitracin. If the bacteria is sensitive to the antibiotic bacitracin, we say that it is positive for Streptococcus pyogenes, which is group A strep here. If the bacteria is resistant to bacitracin, then it is another Streptococcus, but it is not Streptococcus pyogenes. Any question about that? So other Streptococcus are resistant if it is, are sensitive, resistant to bacitracin. And Streptococcus pyogenes is sensitive to bacitracin. Another organism that can be isolated from the throat that's clinically significant is Haemophilus influenzae. This is a gram-negative coccobacillus. Haemophilus influenzae is common ear, eye, and or sinus infection in babies and young children. It is fastidious, but it cannot lyse the red blood cells of a blood agar. And so you have to grow it on chocolate agar, shown here. Chocolate agar is blood agar where the blood is added and the agar is quite hot, and that lyses the blood, and then it turns brown in color. And that's why it's called chocolate agar, because it kind of looks like chocolate. Anyways, Haemophilus influenzae can grow on chocolate agar. It cannot grow on blood agar. Here, we're seeing where they streaked one single streak of Staphylococcus aureus on the blood agar. Let me blow this up. And then they streak this way with Haemophilus influenzae. And here you can see that Haemophilus influenzae is not growing on the blood agar in this region of the plate, but it is growing in a narrow region, a band around the Staphylococcus aureus. Why it can grow here is the Staphylococcus aureus lyses the red blood cells, and that releases the nutrients for Haemophilus influenzae. And so Haemophilus influenzae can grow in this region around Staphylococcus aureus. Any question about that? And if you want to read a little bit more about what's going on, the Staphylococcus aureus is releasing NAD, which is V factor, and hemin, the X factor, from the blood. And then the V factor and the X factor are what are required for Haemophilus influenzae to grow, at least on a blood agar plate. And the chocolate agar doesn't have that problem because it's released already. All right, any question about the cultures grown from the throat? If not, let's talk about cultures grown from urine. Urine cultures are used to diagnose urinary tract infections or UTIs. Bacteria can travel from the urethra to the bladder. And females have a smaller urethra, so they are predisposed to developing UTIs. And UTIs are much more common in females than in males, simply because females have a shorter urethra. To collect a urine culture, the genital area has to be clean prior to collection. Usually you use a clean catch midstrand, involving collecting only from the middle portion of the urine stream. This reduces the cultivation of transient organisms that come from the urethra, because there's bacteria and other microbes in the urethra. You can also get urine cultures from catheterization. It's cleaner, but it is a more invasive process. And then the cleanest process is directly placing a needle into the bladder and then using suprapubic aspiration, as shown in this picture here. You can use suprapubic aspiration, and then your contamination will be essentially nil. Okay. You can also use a catheter, but the catheter itself can pick up bacteria in the urethra and then put it in the bladder to be sampled. And then you can also use a clean catch system, and that is you sterilize the genital area, not sterilize, you clean the genital area, and then you have the urine stream and you catch the urine midway through, not at the end, not at the beginning of the stream. And that makes it less contamination of microbes from the urethra. And that they call clean catch midstream. Any question about that? All right, once the urine is received in the lab, you plate 0.01 milliliters, that is 1/1000th of a milliliter of urine onto both a non-selective media, such as BAP, as well as a selective media that grows gram-negatives, such as McConkey's or EMB plates. You always use McConkey's or EMB or something similar to them so that you will grow the gram-negatives. BAP will grow both the gram-positives and the gram-negatives. And as I stated, most of the urinary pathogens are gram-negatives. And most of them are coliform, such as E. coli. E. coli is the most common isolate in a urinary pathogen, meaning a sample of urine. Here is showing you 1/1000th of a milliliter. You put the urine on that loop, and then you plate that loop over the plate. And that's comparing the amount you're plating to a penny. Don't ask me why it's a Canadian penny. And then you do the same on McConkey's or EMB. The interpretation of a positive urine culture is multifaceted. There are a number of factors to be considered, such as the type of collection. Is it voided, clean mid-catch system versus a catheterized specimen or a suprapubic aspiration? Because only the suprapubic aspiration should give no contamination from the urethra, okay? You can have slight contamination even on that, and that is we clean the skin and then inject the bladder with a needle, but you don't sterilize the skin. But the contamination is very minor on that. It usually is not seen. And if it's seen, it's usually very minor. Some other factors is the gender of the patient, male or female. A male have less contamination from the urethra than females, and I don't know why that is, but that's just the way it is. And then the number of colonies per mil. Like if you get too many colonies and they're polymicrobial, then it may indicate a poor collection. Okay. And it's showing you that there may be a UTI if there's more than 50,000 colonies per mil in a male, but the UTI in a female has to be more than 100,000 colonies per mil. Did I say that right? 50,000 colonies per mil and 100,000 colonies per mil in a female. And why the difference in number, like I said, for whatever reasons, females shed more bacteria from their urethra than males. And we're assuming a clean catch midstream method there. All right, once you have a possible pathogen, you then run different chemical tests trying to isolate what that pathogen is. And you can use the enzyme coagulase to determine if it is Staphylococcus aureus from other staphylococcal species. Other staphylococcal species are catalase negative. Staphylococcus aureus is coagulase positive. What happens? Staphylococcus aureus produces the enzyme coagulase, and then it will coagulate blood or plasma, changing the fibrinogen into a fibrin clot. And this clot around the bacteria can help protect the bacteria from phagocytosis by our immune system cells. And that's why Staphylococcus aureus produces coagulase. And that's to protect itself when the bacteria are low in number. There are two tests to do the coagulase test. You can look at the slide test and you'll just see clumping if or agglutination if the bacteria is coagulase positive, and no clumping if the bacteria are coagulase negative. In the lab, like the unknown project, if you were to ask for this test, I would show you a tube. And if the tube coagulates, becomes semi-solid, that's coagulase positive. And if the tube remains a liquid, that's coagulase negative. Any questions about coagulase? All right. The most common bacteria isolated from urine cultures is E. coli, and it's in the family Enterobacteriaceae. This family is gram-negative bacilli that ferments glucose, and the glucose can be used to separate out Enterobacteriaceae from the other membrane that member of the family member that causes urinary tract infections, Pseudomonaceae, but it's not often used. What we tend to use to separate the Enterobacteriaceae from the Pseudomonaceae is we do the oxidase test. Enterobacteriaceae are oxidase positive. Pseudomonaceae, the family Pseudomonaceae, is oxidase negative. Is that right? No, it's oxidase positive. Enterobacteriaceae is oxidase negative. And Pseudomonaceae is oxidase positive. If the organism is oxidase positive, the oxidase will turn either blue or purple color right here. And this is the area that the reagents have been put, and apparently there was oxidase positive cells there. Pseudomonas aeruginosa, which causes bacterial infections, is oxidase positive. And let me briefly say that it's oxidase positive because it has cytochrome C, or oxidase cytochrome C, in it, which is actually an enzyme in the electron transport chain that we just talked about. And Enterobacteriaceae does not have cytochrome C oxidase, so it is oxidase negative. Any question about any of that? Oxidase negative, you'll put the reagents on over like here or on another plate, and it will be nothing showing up, not bright purple or bright blue. Any questions about the oxidase test? It looks like that's the last test I'm going to talk about. So in summary, urine and throat cultures are two of the more common types of specimens received in a lab for bacterial cultures. You don't need to read about the references or do anything with them. You're not going to be physically performing any of these activities because everything is going to be done online. However, you are responsible for knowing how to perform these activities. So read through them and the enzymes used. We have a video on the catalase test. We have a video on the coagulase test, and we have a video to watch on the oxidase test. There are no virtual interactive labs for any of this lab. And then work on the questions. Don't do it in the lab manual. Work on the questions in the exercise. which also has the link to the videos. And answer the questions. And if you have any questions, I will be here close to 8 o'clock. However, when the last student logs off, I'm going to log off. If the last student is still here at 8 o'clock, I will be here until 8 o'clock. All right, any questions? When you get your answers, fill them in the worksheet and then submit the worksheet to Canvas. This must be submitted by 11:59 p.m. Saturday, or it is late.
Dr. Volpe?
Yeah.
There's a crossed out question. We don't do that one, right? For question
If I have it
8C.
crossed out, you don't do it. Is there one?
Thank
Yeah. 8C, it's not in the lab manual, so I crossed it out. Okay, you don't need to do that one.
Professor,
Uh-huh.
I missed the lecture. I'm sorry, I had an emergency that came up. You are going to post the PowerPoint, correct?
I will post the YouTube video. The PowerPoint won't be posted until we finish the lesson. And we did
Okay.
not finish the lesson today.
Okay,
Okay. It
Thank
will
you.
eventually be posted when we finish the lesson, which it better be Tuesday. I mean, we're supposed to finish it today, but hopefully we'll finish it on Tuesday.
okay, thank
Okay.
you.
All right. All right, if
just
there's
wanted
no
to-
questions, I'm gonna stop the sharing. And hopefully I'll get some grading done. I'm way behind on the grading. But I did send out let me make sure this is being recorded. Yeah, I did send out the unknown numbers. It was very late Tuesday night. In fact, it was slightly after midnight, but I consider that Tuesday night. So I will try to send you the results on Tuesday night. Okay, that's my goal.
Just to make sure, we didn't have to email you like to start those. Just wait for it.
Yeah, after you get the first 3 test results, you will then look at your results, make your interpretations, write up your results and your conclusions. You should also, in your unknown notes page, state which bacteria are under consideration. And then at the end of the unknown notes page, state which test you want to do next.
Okay,
Okay.
perfect.
Notes which I get on that cover page where it says my test number and then say the number that you have on like that cover page on your unknown notes. Are we going to end up submitting the unknown notes more than once, or is this just kind of something for us to keep cumulatively and then we send it to you at the end?
You can do it cumulatively if you want. There's no problem with that. You don't have to. You can just, whenever you update your unknown notes, put a new page there. You don't have to make it cumulative. You don't want it to be. How you do the unknown notes, it's free form. You can do it different ways. If you want to do it very formally, the unknown notes page can be done very similar to your unknown report.
Okay.
The formality of the unknown notes is not required.
Okay. Because it said add your number to your unknown notes because you can't send test results without knowing your numbers. So
Yeah, if you ask for a test and you have no unknown number on your unknown notes page, I can't send you the results.
Okay.
Okay.
Thank you.
On and off, on and
There we
off.
go. What's your question?
Okay, so the unknown notes, just like Ariel was saying, so last night or whatever night, after you posted the numbers, I went into the unknown notes, hit submit assignment, put my name and my number in there. So after that, do I just keep adding to that and hit submit assignment each time? Or do I put this on a Word document or a Google Doc and then I submit it to you all at once?
I don't remember how I set that up. Were you able to just type in there your unknown number and then submit that? Or did you
So
have to-
it's,
Or did you have to
it's
put it in
based-
a document and then submit the document?
no, we could do text only box. So that is available and that's what I did and I submitted it. So
Okay,
I
that's
guess,
fine.
okay, great. I just want to make sure. Okay.
Yeah. So you can do it either way. I didn't remember if I had set up text box in the unknown note pages or not.
Yes, you did.
Okay.
Okay.
All right.
That's how we're going to be requesting from you our future tests, right? Is
Yeah,
through
in the
that.
unknown notes page. Just
Okay.
make sure you make the request at the end of the unknown notes page. Very rarely, I have a student who makes the request in the middle of the unknown notes page, and it's very hard for me to find if it's in the middle, okay? So put it at the end of your unknown notes page, and then I'll see it right away. Obviously, you don't need to do that until after I send you the first test results. Okay?